Actually, there is a few yeast-like organisms that is usually encountered in the clinical laboratories. The first one is Candida. Candida have pseudo-hyphae, and some species show the true hyphae, and blastoconidia also. The organism like Candida can be differentiated on the basis of carbohydrate assimilations and morphology. But like the Candida, the trichosporan and blastoschismomyces also have pseudohyphae and the property of true hyphae production, and also some of these species produce the blastoconidia, but they can be differentiated on the basis of biochemical test, and some annealing conidia. Like if you see the trichosporan, unique properties is that it is also it have the ability to produce the pseudohyphae, true hyphae, blastoconidia along with hyphae, but also the unique property is urease positive. And blastomyces have also the pseudohyphae, true hyphae, and blastoconidia, but the unique property is annealoconidia production, which differentiate this entire group from the rest of the yeast-like organisms. Like the second one is Rhodotula. Rhodotula produce the red color colonies on SDA at 30 degrees centigrade, and also urease positive organism. Cryptococcus is also urease positive organism, and it have a capsule, so and white mucoid colonies on the subrod agar, which differentiate this organism from the rest of the yeast-like organism. Saccharomyces cerevisiae have unique property of ascospore production, which can be visualized on modified acid fast staining, and they also need olive oil in the small amount for their growth enhancement. Like the Malaysia have unique property of sporangia production, and some species of Malaysia, like Malaysia furfur, need olive oil, a fatty acid, for their growth. Prototica is also Prototica is a sporangia production is there, which is the unique property of Prototica, and they also is requirement is they can grow in the presence of a cyclohexamide, and they need olive oil. Gotychium is the unique property of Gotychium, they do not produce the pseudohyphae, which differentiate this organism from Trichosporan, Blastoschismomyces, and Candida, and also produce the true hyphae, and these true hyphae is further. Now we will explain all these yeast-like organisms further in detail. The first one is Rhodotorola. Is sometimes the capsule is there in some species and commonly known as a contaminant, but rarely cause the fungemia, rapid mature in 4 days. And on Quon meal 2080 agar at 25 degrees centigrade, orange to red colonies is a yeast-like budding cells are round to oval or elongated, as seen in the picture. Sporoblomyces salminicore is an environmental source and not grow at 37 degrees centigrade. Its optimum temperature is 25 to 30 degrees centigrade. And white pink color colonies on SDA, and the colony resemblance to the Rhodotonella species, and satellite colony eventually form around the original colony due to the production of blastoconidia, and blastoconidia can be best demonstrated by taping an inoculated cornmeal agar plate face to uninoculate cornmeal plate. After the inoculation at 25 degrees centigrade, the blastoconidia that are shoot off will form the mirror image of the colonies, and the blastoconidia is kidney shape, as shown in the picture. The Saccharomyces cerevisiae is usually involved in the fungemia, and the cream color colony on SDA infection can also occur if the patient have take the probiotics, and probiotics contain Saccharomyces cerevisiae, and it is rapid mature in 3 days. Characteristic round ascospore, which differentiate this organism from the rest of the yeast-like organism. 1 to 4 per ascospores are there, but best demonstrated when the organism is grown on the special medium like V8 acetate ascospore agar, and goro Gorodkova medium, and ascospore stain and canyon stain. Additionally, ascospores are given gram negative shape, while the vegetative cells are gram positive. You can Wicker hemomyces anomolus, they're also called Pichia anomolus and Hansenola anomola. Actually, it is the sexual state of this organism, and Candida pelliculosa is the asexual state of this organism. When culture on the ascospore medium, the kidney canyon or ascospore stain shows 1 to 4 ascus per ascospores, and each ascospore have the brain that give it the helmet or head shape that differentiate this organism from rest of the species. And on the cream color colonies on SDA, and Prototheca species commonly classified as the Echlorophyllus algae, but they cause the mycosis like disease, cutaneous, subcutaneous, and systemic protothecosis. And P. Vickerlami and P. zophi have been involved in the human diseases. It is rapid mature in 3 days. Optimum temperature is 30 but cannot grow on 37 degrees centigrade, inhibited by the cyclohexamide. Sporangia is the unique characteristic of this organism for the various size and contain the ascospores, no hyphae, no budding yeast. These are the Sporangia. Trichosporan species is a normal flora of skin and normal flora of nails, mouth, moderately rapid, mature in 5 to 7 days in immunocompromised and neutropenic person. Also cause white pedria, hair infection, usually caused by the pedria horti. True hyphae and pseudo hyphae with the blastoconidia singly or in chain. And because of the sharp because of the shape of antigen sera from the patient with the disseminated trichosporan infection may give the positive reaction with the cryptococcal antigen latex test. And on the cream color colony and powdery crumb-like filamentous fungi, these are also called monomeric molds form, and they some classification like zygomycetes and diatomaceous fungi, dermatophytes, and hyaline hypomycetes are categorized in this group. Further, these all organisms are opportunistic fungi, opportunistic pathogen. The first one is Rhizopius, which belong to the zygomycetes. It's also called glomerulomycosis and glomerulomycetes or zygomycetes, monomeric molds, and non-septate, inhibited by the cyclohexamide, rapid mature in the 4 days. US split is usually white for all the zygomycetes. And for Rhizopus, it's a commonly laboratory contaminant but can cause the fetal diseases once in immunocompromised patient. It mature in the 4 days. Lid lifter, front is dense, cottony, candy-like growth, white at the first and turning gray or yellowish and brown. And the reverse is usually white. And the hyphae or large broad non-septate produce the horizontal stolon that are attached to the rhizides, and sporangiophore is cluster opposite to rhizides and terminate in dark and round sporangia containing sporangiophore. Mucor species also belong to the zygomycetes, and but the only difference is that they don't have the rhizides, and the sporangia may be branch. Further, this whole group zygomycetes we will explain one by one. Glomerulomycetes class of fungi are inhibited by cyclohexamide and that are encountered in the clinical laboratory belonging to the order Mucorales. Infection due to the fungi in this order can correctly be referred to as mucormycosis, but misinterpretation that they are all caused by mucor species must be avoided. In glomerulomycetes or zygomycetes, Rhizopus species is the most common etiological agent in zygomycosis. The maximum growth temperature is 45 to 50 degrees centigrade, and the genus is differentiated from the mucor species by the presence of stolen rhizoids and usually unbranched sporangiophores, and it is differentiated from the Lysithymia Epsidia species by the location of rhizoids in the relation to the sporangiophore, and by the size and the shape of the sporangia. Lectemia cormibifora complex is formally named as Bisidia, and the maximum growth temperature is 45 to 50 degrees centigrade. It is similar in the structure to the Rhizopia species, except that the sporangiophore of Lectemia species arise often in the small cluster at the points on the stolon that are between the rhizoids. If you see the picture, you can understand this one. The rhizoids are often difficult to detect, and they may best observe by using the dissecting microscope to examine the colony on slanted agar surfaces. Apophyomyces elegans are usually acquired by the traumatic implantation, such as the accidental injuries, surgeries, insect bite, or contamination of burns, and the maximum temperature for Apophyomyces elegans are 42 degrees centigrade, and Apophyomyces elegans is similar like Epsidia or Lectemia, but differ by having the columella is a half circle, sporangia, or pear shape, or periform. A pronounced apoplosis, which is bell shaped rather than conical, a foot cell at the base of the sporangiophore and rhizoid may be between the points of the origin of the sporangiophore or opposite the sporangiophore, depending on the medium. On plain agar SDA, the rhizoid develop opposite to the sporangiophore and fail to sporulate radially with the routine culture method, resistant to cyclohexamide, and reverse plate is pale yellow, which differentiates this organism from the rest of the zygomycete, which reverse plate is white. Saxina visiformis also belong to the zygomycetes. Infection occur in immunocompromised by the traumatic implementation. Infection reported as cutaneous, subcutaneous, osteomyelitic, and rhinocerebral. As systemic infection is often fetal. It is a rapid grow in 4 days up to 45 degrees centigrade. Temperature can survive by this organism, and routine media organism does not sporulate. Non-septate branch hyphae and branch hyaline hyphae form, special known to occur on chespeck agar after 7 days incubation at 37 degrees centigrade. Sporangiophores are flask-shaped rhizoids from the near the base of the sporangiophore are dichotomously branched and darken with the age. And Cochromyces recurvitus is a genitourinary site organism that causes the cervix, vagina, and bloodstream infection. Mature in 5 to 10 days. Optimum temperature is 37 degrees centigrade. Survives up to 42 degrees centigrade. Thermally dimorphic colony. First cream to 10, then feet-like or powdery. Reverses tend to brown and preferably on enriched media such as the brain heart infusion agar in 5 to 7% carbon dioxide from gray to slightly wrinkle yeast-like colony develop into 2 days. Spherically septate hyphae are seen, and the sporangiole contain 12 to 20 sporangiophore that are smooth-walled and of variable size, shape, and no rhizoids are seen at 37 degrees centigrade in 5 to 7% carbon dioxide. Thin wrinkled round yeast develop which single or multiple budding that may resembling the ship wheel appearance or of Paracoccidia brasiliensis shape. You can see the infection 37 and 25 degrees centigrade. It is the only dimorphic fungi that is categorized in zygomycetes. And the most interesting and unique characteristic of Saxina visiformis is a branch hyaline hyphae. Here the Rhizopia species can be seen on SDA slant at 30 degrees centigrade after 4 days. Note the robust cotton candy-like growth, typical of many zygomycete sardorum. Mucorales. Lithimica epsidia, formerly called epsidia, have also cottony candy-like growth. And if you see the Coxromyces in PDA and Conangamella bertollerate slant, it's also like as a Rhizopia species. Conangamella bertolleretia is an uncommon cause of cutaneous infection followed by the traumatic inoculation, occasionally been involved in the pulmonary and disseminated infection in immunocompromised. It is rapid, mature within 4 days, can grow at 40 to 45 degrees centigrade, broad hyphae, almost non-septate, and the vesicles are covered with the spine-like denticles, each supporting a round to oval sporangiola, are often encrusted with the needle-like crystals, and rhizid zygospores, chlamydiospores may be seen.